In today's video, we're going to talk all about forensic anthropology. What can we learn from bones? So a little bit of the historical development of the field. In the 1800s, scientists began studying skulls, and this laid the framework for today's knowledge. Now, a lot of this analysis performed during this time was not super scientifically accurate. Things like how bumps on a person's head could predict the kind of person they were. But it was the first time that we started really looking at skulls and sort of saying, I think these are really important to who a person is. In 1932, the FBI opened their first crime lab, which did involve skeletal remains. And the Smithsonian Institute became its working partner in the identification of human remains. So the FBI does not deal with the identification of human remains itself, but it does use the Smithsonian Institute for that. And soldiers killed in World War II were kind of the first big test case for using forensic anthropology techniques to identify remains, and many deceased World War II soldiers were identified using these techniques. So how do we actually get bones? So bones originate from cells called osteoblasts. These osteoblasts migrate to the center of cartilage production and deposit minerals. So when bodies are developing, they have a lot of cartilage, making them very soft and squishy. If you've ever met a baby, they're soft and squishy. And osteoblasts go to that cartilage that's much squishier than bone and start depositing minerals into it, turning cartilage into bone. Throughout life, bones are being broken down, deposited, and replaced. Our bones are a living part of our body that is maintained by our body. Osteoclasts are the second type of bone cell, and they are partly responsible for this maintenance of bones. They do a lot of different things, but mainly remove cellular wastes. How I remember them is osteoblast, B, comes before osteoclasts, C. B comes before C. We can't have a cleanup crew until we have bones to begin with. So how is our skeleton really all connected to each other? So bones are held together by cartilage, which wraps around the ends of our bones and keeps them from scraping against one another. This cartilage can become broken down, and it is incredibly painful when bones start rubbing against each other. We have ligaments, which are bands that connect two or more bones together. This is why if you pull on your arm or your leg, the bones don't come apart. It's because ligaments are holding them there. Then we have tendons, which connect muscle to our bone. So most of our bones are wrapped in muscle, so that muscle is being held on to the bone by tendons, which is why you can't just move the muscle out of the way. Until about 30 years of age, your bones are increasing in size. They are continuing to deposit minerals, and so what you eat, drink, all of that is really influencing what your skeleton is going to become. So technically, after 30 years of age, your skeleton stops increasing in size, and then you enter maintenance mode. So to maintain your skeleton, it's important to keep eating a healthy diet, to continue exercising, because we're trying to slow the deterioration of our skeleton through these processes. So what can bones tell us? So an osteobiography tells much about a person through the study of their skeleton. Now this makes sense because the prefix osteo means bone, and you know that biographies are the story of someone's life. So it's a bone story of someone's life. Now there are way too many things to get in the course of this video of all of the neat things about the human skeleton, but one of the ones that I think is the coolest is the bones of a right-handed person, for example, are slightly larger than the bones on their left arm. So a skilled forensic anthropologist could tell whether someone is right-handed or left-handed solely based off of the bones of their body, because their right arm bones are going to be slightly larger than the left, and vice versa if the person is left-handed. I just think that's so neat. Forensic scientists essentially realize that bones contain a record of a physical life. The things we eat, drink, the harm that comes to us can often be reflected in our skeleton. So by analyzing bones, we can reveal clues to things such as sex, age, height, and even overall health. So sex determination is one of the most common things to do with skeletal remains, and it's also pretty straightforward. So talking about skulls first, in the male skull, they typically have more square eye orbits. Your eye orbits are the indentions into your skull in which your eyes sit. Meanwhile, female individuals typically have more rounded eye orbits. Male skeletons tend to have a square mandible. This is often why we refer to men as having squared off jaws, and it's because their mandible, the lower part of their jaw, literally is more squared off, compared to female skeletal remains that typically have a more V-shaped mandible, which is why we refer to women as having delicate facial features, because that V is much more delicate. A thick, large brow ridge. So your brow ridge is right about where your eyebrows sit on your skull. And in many males, there's actually more bony growth there, causing it to protrude a little bit, compared to women have a much thinner brow ridge. So while it may protrude a little bit, it's typically not very much. The occipital protuberance is a bump on the lower back of your skull, just above where your neck connects to your skull. You can look up the exact location online if you would like. Men have one, and women typically do not have one. As far as foreheads are concerned, male skeletons typically have a low and sloping frontal bone. That's what this V right here is showing you, how it's much lower compared to females, which have a much more high and rounded frontal bone, which would be your forehead and into your hairline, once again, as you can see in this picture here. And one of the ones that I think is the most interesting is the surface of male skulls is typically kind of rough and bumpy, and that the typical female skull is much smoother. This has a lot to do with the muscle connection. Male faces tend to be more muscular, and those muscles are actually leaving little indentations on their bones, versus the female face does tend to be less muscular, meaning it's not connecting and leaving those marks on the bone. Now, something important to note. Not all male skeletons will have every single one of these facial characteristics. Similarly, not all female skeletons will have every single one of these facial characteristics. It is possible to kind of mix and match, but we take kind of an average of what the facial features are to determine male versus female. Now, the bone that is the absolute best for doing a sex determination is the pelvis. The reason that this is going to be the best bone is, unlike the face that really has a lot to do with appearance as far as sex difference is concerned, the pelvis has to do with reproduction. And because of the different roles of males and females in reproduction, their pelvises are very different. So therefore, it is much easier to tell a male pelvis from a female pelvis as opposed to the skulls. So males will have a less than 90-degree subpubic angle, and you can see that occurring right here. It's a pretty narrow angle compared to females that are going to have a 90-degree or greater subpubic angle, and as you can see, it's right there. Now, this is because baby is going to have to pass under the subpubic angle when giving birth naturally, so we need a wider opening there. Additionally, males tend to have a triangular pubis, while females have a rectangular pubis, making their pelvis more stable, because essentially baby sits in pelvis for the time it is inside mother. Males have a heart-shaped pelvic cavity, that is the opening inside the pelvis. Now, it is not a true heart, but it does kind of look heart-shaped when you look at it, compared to females that have a oval-shaped pelvic cavity. Now, baby does have to pass through the pelvic cavity, so an oval is much more conducive to a baby going through compared to a heart-shaped. Then males have a long, narrow, and curved inward sacrum. Your sacrum is also known as your tailbone more commonly. So in many men, it is longer, thinner, and also kind of curves onward, as opposed to females that have a short, broad, and curved outward sacrum. And once again, this has to do with childbirth and not wanting baby to break tailbone during childbirth. So you can see that the differences are much more stark, which makes it a much better tool to use if we have the pelvis available to us. Now, we can also tell the age of a skeleton through a few things. One of those is cartilaginous lines. So during life, we are originally born with 450 bones. However, by the time we reach adulthood, we only have 206 bones. So we have many bones that were once separate that joined together throughout our life. So originally, we have bone being separated by cartilage, and over time, those osteoblasts, which we previously learned about, are going to replace that cartilage with bone, and as it does so, we get what we call an epiphysis line that is visible. When the cartilage is fully replaced, that line is no longer visible. So we can track where that line is, how visible it is, or if it's no longer visible to determine an approximate age of someone. So this information can be used to approximate a skeleton's age. Something else we can use are called suture marks. So your skull is full of a variety of suture marks. When you were born, your skull is still separated into multiple separate plates. This is why we often refer to babies as having a soft spot, because there literally is a spot on the top of their head that does not have any bone over it. However, throughout life, these plates begin to fuse together and leave behind what are called suture marks. So by about age 21 to 30, the lambdoid suture will have closed. That is the suture right across the back of your head. By about age 30 to 32, the sagittal suture, this is the one going from the front of your head straight to the back, will have closed. And then last but not least, between 48 and 50, the coronal suture will have closed. And that's the one that's kind of like a headband going from ear to ear, or where a crown would sit, hence the name of coronal. So we can look at a skull and see how visible or how put together these sutures are to get an approximate age of an individual. Another very popular way of aging skeletal remains is by using the teeth. So as human beings grow up and get older throughout life, teeth grow, change, and pass through different stages. Each of these stages has certain unique characteristics which can help to determine a person's approximate age. In some cases, the tooth stage is visible to the naked eye, while in other cases, it is necessary to have a closer look under the microscope or even to do an X-ray. So once a tooth erupts, breaks through the jawbone, it starts to build 2 layers of hard tissue called the cementum around its root every year, trying to keep your teeth strong and in your mouth. If a cross-section of a tooth is examined under the microscope, these layers appear as light and dark rings, very similar to tree rings. I know it's really weird to think about, but your teeth are like trees inside of you. So scientists can count the number of layers, and if they add the eruption year, so the age that tooth would have erupted, and if they don't know, they can kind of do an estimate based on the average age those teeth come in. They will get the person's age at death. Scientists can also perform special chemical tests on teeth to determine a person's age. They can also sometimes use these chemical tests to determine where a person may have been from based on the mineralization of their teeth. So just to give you a look at what I'm talking about, we can take this tooth right here and cut it in half, and you can see that when you look on the inside, you do see these rings. This is an electron microscope. You can see the very detailed look at these rings. So we would say that they got in that tooth at age 10. We counted 33 rings, so that person is approximately 43 years old. Something else you can calculate from a skeleton is the height of that individual. So just as the age can be estimated by looking at the bones of the arm or leg, we can also estimate height. Often the approximate height of a person can be calculated from one of the long bones, even if just one is found. Our long bones are our humerus, the upper arm bone, and our femur, our upper leg bone. However, you can use the tibia, the fibula, the ulna, or the radius, the 2 bones in your lower leg and the 2 bones in your lower arm, respectively, in order to make these calculations as well, but the humerus or the femur are going to be best. Sex and race do need to be taken into consideration when making these estimates, so it is important to have done those estimations first in your skeletal analysis before estimating height. Now, there are formulas that are used to make these estimations. I am not asking you to memorize those formulas, but you will be working with them in lab. Something else we can determine about an individual is their ancestry. So determine for ancestry of skeletal remains is probably the most difficult one we're going to talk about today. This is for a variety of reasons. Different ancestral or racial groups do have many physical similarities in common that are seen on skeletons. Also, not every person has the characteristics associated with their ancestral group. So this is probably the most difficult. But some things that we can look at are the shape of the eye sockets, Are they oval? Are they circular? Are they square? These tend to vary between different ancestral groups. The absence or presence of a nasal spine. Some racial groups have nasal spines, others do not. The nasal index, which the nasal index is the ratio of the width of the nasal opening to the height of the opening times 100. So different ancestral groups have different average nasal indexes. Proganthism. Proganthism is about how far your upper and lower jaw essentially are apart from each other. So this is the projection of the upper jaw beyond that of the lower jaw. Now this is different than what we would call an overbite. This has to do with the actual shape of the face. The width of the face. Some racial groups are known for having wider facial structures than others. And also the angulation of the jaw and face. Some racial groups are known to have more softer angulation, while other racial groups are known to have more pronounced angulation. So as I said before, these are pretty difficult, but you are going to get to try your hand on it in class. Now, something that can be done if we do have a skull of skeletal remains and we're trying to figure out who they are is a facial reconstruction. So your face is formed by the skull and the muscles and tissues on top of the skull. This is why your face is unique to you and only you, because your skull is actually unique to you and only you. So a face can be rebuilt from just skeletal remains. Now, facial markers are positioned at critical locations on the skull, and then clay is contoured to follow the height of the markers. So we know approximately how thick certain muscles are in different locations of the face, so we start with that. Now, while sometimes this is still done with clay and markers, a lot of times today computer programs are performing very similar functions of taking that skull and rebuilding the face virtually instead of with clay. This has a lot of advantages. Most importantly, the time it takes and the ability to make changes. On a computer, you can make changes very quickly. If you need to change the eyes from blue to brown, for example, this is a very detailed process if you're using clay. It's a very simple process if you're using a computer. Now, these computer programs were originally developed to age missing persons or criminals. You may have seen the Have You Seen Me? And they were last seen in 1993, so they aged them up 20 years, 30 years to see what they would look like today. That is the same technology. Now, I know as you've been watching this, some of you may have been wondering, why are we putting in all this work to identify skeletal remains? Can't we just do DNA? So bones actually contain very little nuclear DNA. And when we say DNA, we're typically talking about nuclear DNA, the DNA that makes you, you. However, like hair, bone does contain mitochondrial DNA. Now, you may remember that this is DNA that is inherited only from the mother. So it is not unique to you and only you. It is the same as everyone in a maternal line. However, the advantage is that long after nuclear DNA has been lost through tissue degradation, that mitochondrial DNA can still be obtained from the bone because bones last a very long time. We can compare the results to living relatives on the mother's side of the family to identify these skeletal remains. But once again, mitochondrial DNA is not unique to an individual person. It will match everyone on a mother's side of the family. Now, last but not least, once we've kind of determined age, sex, height, all of these important things about an individual, like a medical examiner, a forensic anthropologist can also look at what might have happened to this person by performing a skeletal trauma analysis. So forensic anthropologists often determine if damage to the bones occurred before or after death. This is important to determine which injuries occurred before death. If so, they can even determine how long before death they may have occurred or after death. Once again, important in determining whether these injuries were just the part of body disposal or whether these actually happened to a living person. Definite distinctions exist between patterns on the bones made by weapons and patterns created by the environment. So we can tell blunt force trauma from like a baseball bat versus a gunshot wound, even on the skeleton, versus animals or the environment having an effect on the body while it's in the wild. Sharp force trauma, blunt force trauma, gunshot wounds, and knife wounds all leave distinctive patterns if they hit bone. That is the important part. It is possible to injure someone without injuring their skeleton, but a lot of times there is injury to the skeleton, and we can use that even if we no longer have any tissue to help figure out what happened to this person. So even if we only have skeletal remains left, we can still figure out who we have and what happened to them. And with that, I hope you come to class with any of your forensic anthropology questions.